Anopheles gambiae, the mosquito behind most of Africa's malaria. This week, researchers published something uncomfortable about it. In the proceedings of the National Academy of Sciences, a team reported striking levels of insecticide resistance in this vector, and it wasn't one mutation doing the work. Several different molecular mechanisms were acting together, synergising. The whole was worse than the sum of its parts. That matters, because the front line against malaria is still a piece of cloth soaked in pyrethroid. When resistance stacks, a net stops being a barrier and starts being a nuisance. One of those mechanisms has a name, knockdown resistance, a tiny change in the sodium channel that pyrethroids are supposed to jam. Change the lock and the key stops turning.
A new review traced how that change has evolved in Aedes aegypti, the yellow fever mosquito, genotype by genotype. Now keep one variable in mind, temperature, because here's the twist. Another study this week showed the resistance genotype changes how temperature affects the poison pyrethroid toxicity is temperature dependent. Which mutations a mosquito carries decides which way that goes. The same spray on the same species can behave differently on a warm night. Now meet Aedes albopictus, the Asian tiger mosquito, daytime biter, container breeder, world traveller. In southern West Bengal, researchers screened tiger mosquito populations for that same sodium channel resistance. What they found was heterogeneity, not a countrywide picture, a patchwork. High in one place, low a short distance away. Control that works in one district can quietly fail in the next. And then there's this bisphenol A, the plastic additive, the one leaching from the discarded containers these mosquitoes breed in exposure to it, strengthened Aedes albopictus tolerance to chitin synthesis inhibitors by acting on an estrogen-related receptor. Our pollution is training their larvae, so the field is leaning harder on biology. Wolbachia is the star, a bacterium that lives inside mosquito cells and sabotages their eggs. But a paper in Molecular Biology and Evolution found that sabotage is temperature sensitive and the weak point tracks the transcription of a single gene, not simply how much bacteria is present. That's the laboratory. Here's a back garden. This summer, about 600,000 mosquitoes are being released across the Washington, D.C. area, deliberately. Every one of them male. Male mosquitoes don't bite people. Those males carry Wolbachia. The same naturally occurring bacterium. When a released male mates with a wild female, the eggs she lays simply never hatch. No poison, no bite, just a dead end. It's called the incompatible insect technique. What's new is who is doing it. Bee Safe Mosquito Control is the first business in the United States to offer this kind of control straight to residents. Until now, this was the work of local government mosquito control districts, public health agencies, and research partnerships, and the scale is about to jump. Alphabet, Google's parent company, is seeking approval to release up to 64 million Wolbachia mosquitoes in California and Florida. 600,000 against 64 million. Why the urgency? Because climate change is creating more favorable conditions for mosquitoes to survive and reproduce. More mosquitoes in more places means more worry about the diseases they carry. Some answers are older than all of this. In Burkina Faso, researchers deployed Toxorynchites brevillepes, a mosquito whose larvae hunt other mosquito larvae and whose adults never bite a human at all. A mosquito used against mosquitoes, same idea, smaller animal, copepods. Tiny crustaceans dropped into household water jars where they eat Aedes larvae for you. A new review mapped how communities actually run these schemes from buckets to backyards. Heat cuts both ways. In Aedes aegypti, exposed to a heatwave, dengue virus too struggled. Vector competence went down, and the reason was a disruption in 5-methylcytosine, a chemical marker on the mosquito's own RNA. Warming doesn't only make transmission worse, sometimes it breaks it. So notice the pattern. 3 separate papers, 3 different systems, one hidden variable. Temperature decides how the poison lands. Temperature decides whether Wolbachia holds. Temperature decides whether the virus takes, and every time the switch underneath it is genetic. A channel gene, a transcript, a mark on RNA. Warming isn't the backdrop to this story, it's an input. Meanwhile, the map keeps moving. Aedes albopictus has now been recorded in Magdalena at the northernmost edge of Colombia. New ground and a new arbovirus risk for the people living on it. Chikungunya is resurging in Thailand in Guangdong. Modelers ask what imported cases could do next, forecasting the risk of retransmission. And a global meta-analysis pooled more than 158,000 tested people to measure how much dengue and chikungunya is really out there. One more vector deserves a moment. The triatomine, the kissing bug. A dermatology paper this week is a reminder that the bite leaves a mark and that a skin reaction may be the first and only warning of exposure to Chagas disease, which is the problem with Chagas. It hides for decades. Then it damages the heart. So a Brazilian team trained artificial intelligence to read the electrocardiogram and pull the signature of Chagas out of a trace a clinician might pass over. All of that came from one single week, 98 papers. The mosquitoes are adapting faster than our chemistry. The good news is, so is the science. This has been Bug Bites. See you next week.